Therapeutic drug monitoring. So what is therapeutic drug monitoring? This is the measurement of specific drug concentrations in a patient's blood, serum, or plasma at scheduled times. So the purpose of therapeutic drug monitoring is to ensure that the drug is effective, or that it's working as intended, that it's safe and not causing harm, and that it's personalized, so that we're giving the right dose for that patient. So we want to be able to optimize the therapeutic effect. The goal for that is to achieve the desired drug concentration that produces the maximum benefit to the patient. So for some drugs, too little drug in the blood is just going to be an ineffective therapy. So an example of that would be a subtherapeutic level of anti-epileptics such as phenytoin may lead to seizures. TDM, or therapeutic drug monitoring, helps to confirm if the patient is within that therapeutic range, which is the concentration range where the drug is actually working, where it's effective. We also want to minimize toxicity. So the goal of that is to avoid drug concentrations that exceed safe levels, which can lead to adverse effects or even organ damage. An example of that would be a high level of digoxin can lead to cardiac arrhythmias. Excess aminoglycosides can actually cause kidney damage or hearing loss. So therapeutic drug monitoring can help provide an early warning system for potential drug toxicity, especially in high-risk patients. We also want to make sure that we can guide dosage adjustments. The goal would be to adjust the dose based on the patient's individual response. And we also have pharmacokinetic variability, which we'll get into, and then drug interactions or disease states. So an example of this would be a patient with renal impairment may not be able to eliminate the drugs efficiently. So the therapeutic drug monitoring will help reduce the dose to prevent accumulation of that drug. Dosing is not a one-size-fits-all. Therapeutic drug monitoring can help enable, individualized therapy with that. Some definitions that we need to know. So we have bioavailability. The definition of that is it is the fraction or percentage of an orally administered drug that reaches the systemic circulation unchanged. IV drugs, because it goes directly into your blood system, has 100% bioavailability. Oral drugs usually have less than 100% due to incomplete absorption or first-pass metabolism. So an example, real quick, is if we have 50 milligrams of a 100 milligram oral dose reaches circulation, the bioavailability is 50%. We have peak concentration, also known as Cmax, the maximum concentration of a drug in the bloodstream after administration. Occurs at Tmax, the time to reach peak concentration, and it can reflect the rate and extent of absorption. So the trough concentration, or the Cmin, is the lowest concentration of a drug in the bloodstream before the next dose is given. It's important in monitoring drugs with narrow therapeutic windows, such as vancomycin or digoxin. It can help avoid subtherapeutic levels or even toxicity. Then we have half-life. I'm sure you've heard of a half-life before. The definition is the time it takes for the plasma concentration of a drug to reduce by 50%. This influences dosing interval and time to steady state. It is dependent on volume of distribution and clearance. So an example would be a drug with a half-life of 8 hours will reduce from 100 milligrams per liter to 50 milligrams per liter in that 8 hours. Steady state definition is the point at which drug intake equals drug elimination, which results in a stable concentration in the blood. It can be achieved after approximately 4 to 5 half-lives of regular dosing, and it is critical for drugs requiring long-term therapy, such as antidepressants and anti-epileptics. And then we have elimination rate. The definition is the rate at which a drug is removed from the body. So it usually follows first-order kinetics, where there is a constant percentage is eliminated per unit time. The zero-order kinetics is a constant amount of elimination, phenytoin and alcohol. This is just a graph that just kind of shows you those definitions instead of just words. So we have a short turnaround time need for therapeutic drug monitoring. So we have the turnaround time is the time from sample collection to reporting of results, which I'm sure you guys are aware of that. But we have rapid dose adjustment. So the reasons why we need this, right? So this is going to be especially important for drugs with narrow therapeutic windows like aminoglycosides or phenytoin. It's going to require a prompt adjustment to prevent toxicity or therapeutic failure. We can also have critical patient status. So in the ICU or emergency rooms, we could have delayed results can obviously cause a serious consequence. We want to avoid toxicity. So high drug levels like digoxin or lithium could require some intervention. So if we're able to get that to them quickly, we can hopefully help turn that around. Early detection of non-adherence, so we can quickly identify if someone has missed their dose, and we can try to help with the compliance on that. And also timely clinical decision making, so for drugs requiring loading doses or adjustments during renal failure, time is of the essence. So an ideal turnaround time would be less than 24 hours, but we usually want to try to get it to 4 to 6 hours if at all possible, or even less would be ideal. So depending on where you work, that's just kind of going to depend. So when to monitor for therapeutic drug monitoring, when you have a narrow therapeutic index drug, so like lithium, phenytoin, vancomycin, digoxin, small differences in dose can lead to toxicity or even treatment failure. When we have suspected toxicity, so when side effects resemble drug toxicity, like if they start to have an altered mental status or something like that. Renal or hepatic dysfunction, so impaired drug elimination requires monitoring and dose adjustment. Non-compliance or adherence concerns, so making sure we're checking those levels so we can confirm if the patient is actually taking the drug and not, selling it or giving it to someone else or saying they're taking it and it's not working, or, something of that nature. Drug interactions, so some drugs can alter the metabolism or excretion of others, like enzyme inducers or inhibitors. And then dose individualization needed, especially in neonates, elderly, or obese patients, because they can have altered pharmacokinetics. So timing, as we mentioned earlier, the trough is the level where it's collected just before the next dose. So it's most common and clinically useful for many drugs, and this will indicate the lowest concentration of the drug. Then we have the peak level. This is collected shortly after dosing. This is going to depend on the drug and what its half-life and stuff is, but it's used to assess absorption and risk of toxicity. So if you have a timed peak or trough drug level and it's not drawn at the right time, this can cause issues. It can cause a misleading result for the patient. So an example, drawing vancomycin level too early, this may appear that the result is too high, and that would be false. So some special considerations, we have steady state concentration that should be achieved usually after 4 to 5 half-lives before sampling. The route of administration can also affect timing if they were given by an IV or orally, and then documentation of the sample time. So when you do collect this, or if you get it down in the lab to run on the chemistry analyzer, a lot of times it's going to be documented in the computer what time it was actually collected, and that is for the doctor or nurse so they can look at it and know if it was collected late or early or on time, and they can make their judgments on that. So pharmacokinetics, this is the study of what the body does to the drug. It describes how a drug moves through the body from the time it's taken until it is eliminated. Pharmacokinetics is often going to be summarized by the acronym ADME, and this stands for A for absorption, so getting the drug into the bloodstream. Most drugs are taken orally and have to pass through the GI tract. After absorption through the gut wall, the drugs will then enter the systemic circulation. Some drugs will undergo a first pass metabolism in the liver before even reaching that circulation. Then we have the D for distribution, so where the drug goes in the body. So once in the bloodstream, the drug is transported to tissues and organs. Some drugs bind to plasma proteins like albumin, which can affect the availability. Lipid-soluble drugs may cross the blood-brain barrier or accumulate in fat tissues. And then we have M for metabolism, so how the body breaks down the drug. This will mainly occur in the liver via enzymes, and then it will convert drugs into the active metabolites, which means that they're still effective, or an inactive metabolite, which means they're ready for excretion. Then we have E for excretion, so how the drug leaves the body. Most drugs are eliminated through the kidneys or the urine. Some can go through the bile or the lungs or sweat as well. So after oral dosing, when a drug is taken orally, it undergoes several key steps. So one is absorption. It begins in the stomach or intestine. The drug has to pass through the GI lining into the bloodstream. And some factors that can affect absorption is, the drug formulation, food intake, your gut's pH, and GI motility. And then we have first pass metabolism. This is the drugs absorbed from the GI tract enter the portal circulation and pass through the liver before it actually goes to the systemic circulation. So some of the drug may be metabolized in the liver, which can reduce its bioavailability. Drugs are grouped into classes based on their mechanism of action or primary therapeutic use. So this helps clinicians choose the right treatment for special conditions. So I'm not going to read these to you, but they are all listed here. Well, not all, but a lot of them are listed there with examples of the type of medication and also for certain uses that they would be used for.